Biotechnology is the use of natural biological systems to create products or achieve an output desired by humans. There are many different techniques used to produce different products in modern-day biotechnology. Cloning can be done at many different levels. Scientists can clone entire organisms, specific cells, or just specific fragments of DNA. Gene cloning is a biotechnology tool to produce many identical copies of a gene of interest. Genes might be interesting because we want to make the protein product that the gene codes for, or to create a genetically modified organism, or to perform gene therapy to alleviate a genetic disease. In the image, genes that code for the protein insulin are being cloned. The gene is taken from a human cell and placed inside a bacterial plasmid. A plasmid is a small extra piece of DNA that some bacteria contain. Once the bacteria contains the insulin gene, it is allowed to replicate. As the bacteria replicate, they produce the protein insulin. This is the product we want to collect to make pharmaceuticals to treat diabetics. When the scientists make the plasmid out of bacterial DNA and human DNA, it is called recombinant DNA. Recombinant DNA is any sample that contains DNA from two different sources. When making recombinant DNA, a vector is used. This is the DNA that is going to be manipulated. In this case, it's a plasmid. Plasmids are small and circular. They are not the main chromosome in bacteria, and they are capable of replicating on their own. Getting the gene of interest into the plasmid requires two different enzymes. The first is a restriction enzyme. It is used to cut both the plasmid and the gene of interest. These are called restriction enzymes because they chop up DNA and restrict viruses from infecting bacterial cells. When the restriction enzyme cuts the DNA, they leave distinct edges called sticky ends. In a sticky end, one of the DNA strands hangs over further than the other strand. The second step is to bind the pieces of DNA together using the enzyme DNA ligase. The sticky ends make it easy for the new gene to be inserted into the plasmid vector. Once the DNA has been sealed, it is now recombinant DNA, and it is taken up by the bacterial cells. Then, when the bacteria cells replicate, they also replicate the recombinant DNA, making many copies of the inserted gene. DNA sequencing is the process of determining the order of nucleotides in a DNA molecule. This tells the scientist the order of the bases, if it is adenine, guanine, cytosine, or thymine. Knowing the sequence of DNA is an important first step for many biotechnology applications. It can be used to identify the genes that cause disease and then treat them, the study of forensic biology, and understanding evolutionary relationships among organisms. The technology used to sequence DNA has improved a lot since it was invented in the 1970s. Now, color dyes are attached to each DNA base. A different color is used for each one. In this example, the dyes were green for A, adenine, red for T, thymine, and blue for C, cytosine. Usually we are only sequencing a small portion of DNA at a time. Since DNA is such a small molecule, in order to sequence it, scientists need many copies of the gene that they want to sequence. So before sequencing the DNA, they have to make copies of it. The polymerase chain reaction, or PCR, is a very fast way to make billions of copies of a gene. PCR amplifies DNA, which means to make many copies. PCR is very specific and copies a targeted gene of interest that is usually very short. PCR works by going through a series of cycles. During the cycle, the sample is heated up to break apart the double helix of the target DNA. Then a primer is placed on both ends of the gene to be copied. Finally, a heat-resistant DNA polymerase enzyme adds new DNA nucleotides to the DNA strands using complementary base pairing. At the end of each cycle, the amount of DNA is doubled. After the first cycle, one strand of DNA became 2 strands of DNA. Both of these are then copied into 4, and then 8, and so on. This is exponential growth, and it is very fast. If a sample is started with only one DNA strand, It would only take 20 cycles to reach over 1 million copies, and only 30 cycles to get to 1 billion copies. That's a lot of DNA. After a gene has been copied using PCR, it can be analyzed in many ways. An early method of DNA analysis was gel electrophoresis. First, the sample DNA is treated with restriction enzymes that cut it up into fragments. Then the cut up DNA is placed inside the gel. The gel is submerged in fluid and an electric current is run through the gel. DNA has a slightly negative charge, so it is repelled by the negative end of the current and attracted to the positive end of the current. This makes the DNA move through the gel in a straight line away from the well. It is easier for small fragments to move through the gel, so they travel further. The bigger pieces travel slower and don't go as far. This allows us to take a piece of DNA and see the sizes of the fragments. When all of these fragments are dyed, they create a distinct banding pattern that acts as a DNA fingerprint for that sample. The sample can be compared to other samples to see if it contains the same DNA. Now, modern-day analysis tends to use short tandem repeats, or STRs, to analyze DNA. Short tandem repeats is when DNA has very short sequences, like GATA, that are repeated several times next to each other. STR analysis doesn't require restriction enzymes. The STRs can be labeled and sequenced using automated technology. STRs are useful because they are different between different people. Each individual has a certain number of repeats at each locus. In the image, the top gene has 4 repeats and the bottom gene has 6 repeats. The number of repeats of STRs allows scientists to compare genes between different people. Both of these tools, gel electrophoresis and STR analysis, can be used to build DNA profiles. A DNA profile is a distinct DNA fingerprint, and each person's will be different unless they are identical twins. These are especially useful in forensic biology used to help solve crimes. In the example, DNA blood evidence was collected at a crime scene and compared with 2 main suspects. Both the gel electrophoresis banding and the STR analysis match those of suspect B and not suspect A. This might be enough evidence to convict suspect B of the crime. The Innocence Project was founded in 1992 and works to help free convicted criminals who are actually innocent. New DNA technology can be applied to older cases to exonerate people who were wrongfully convicted. Earl Washington is one example of an innocent man released from prison after being wrongly convicted. He was convicted when he was 22 in 1984 and released in 2001 after serving 17 years in prison, many of which were on death row. He came within 9 days of being executed. He was awarded almost $2 million by the state and inspired a new Virginia law that allows criminals on death row to ask for DNA testing at any time. Genome editing is a relatively new DNA technology. DNA editing means targeting a specific gene to be replaced or removed, and thus editing the genome. CRISPR is the most common gene editing tool. It was discovered by 2 women, Emmanuelle Charpentier and Jennifer Doudna, who both received the Nobel Prize in 2020. They published their work in 2012, and the use of CRISPR has exploded in the decade since. The CRISPR system is based on an enzyme called Cas9 that can cut through DNA. Cas9 identifies the correct gene to be cut because it is given a guide RNA that is complementary to the gene of interest. The RNA helps the enzyme find the right spot, and then the Cas9 can cut through the DNA. Because the RNA guide can be manufactured and changed, this system can be used to target almost any gene in any organism. It is most often used to do DNA knockout studies. These are studies where scientists are trying to figure out the function of specific genes. They use CRISPR to knock out or break the gene so that it doesn't function. Then they can examine the organisms to see how they are affected by the missing gene. Another use of CRISPR is to insert a piece of DNA into a gene. After the DNA is cut by Cas9, a new piece of DNA can be inserted to change the DNA. This is still being tested to treat disease, Imagine if humans with genetic diseases could be cured because you could remove the mutation and insert the correct unmutated gene sequence with CRISPR.